गांधी सर, ओके।
ओके सर, ओके।
Good morning, dear participants. We are waiting for the guests. Once he arrives, we'll start the session. Good morning, sir. Good morning, dear participants. Dibakar, sir, am I audible?
Yes, sir.
Dibakar, sir,
Network
Hello, sir. Diwakar, sir. Good morning, sir. Sir, you are not audible. Your microphone is not working, sir. Hello, Diwakar, sir.
Yes, yes, I'm here. Yeah,
Yes, sir. Are you able to hear me, sir?
yes, sir. Yeah, I can hear you.
Yes, sir. Thank you. Yeah. Shall we start, sir?
Yeah, we can start.
Very good morning to all the dear participants. Welcome to the third day of national level online training program, which is being organized by School of Pharmaceutical Sciences, Sandeep University. The first two sessions were really fruitful. The audience are enthusiastic. They have asked so many questions, queries, and we had almost 40 minutes of discussion in first day and 1 hour of discussion in the second day. And one more interesting session is awaiting for you. We have renowned speaker with us, Dr. Divakar Selvaraj sir, who is working in JSS UT, and he is going to present very interesting topic on network pharmacology. Before starting our session, let me take pride in inviting our faculty member, Abhishek sir, Department of Quality Assurance, to kindly introduce today's guest.
Good morning, all. So today, my privilege is to introduce Dr. Divakar Selvaraj, who has an enormous experience in Department of Pharmacology. He is currently working as a lecturer in JSS College of Pharmacy, UT. He has more than 5 years of experience of research and 10 years of experience in teaching. He has got numerous fundings. To name a few, ICMR, he has received a ICMR grant of 29 lakhs almost as a principal investigator, where he has worked on a design, synthesis, in silico and neuroprotective evaluation of ropinirole derivatives for the treatment of Alzheimer's diseases. He has also worked on a project as an SRF on molecular docking and selection of ethyl ferulate as a potent capable inhibitor to activate the NRF2 pathway for the treatment of Parkinson's diseases. He has got many publications having H index of 12 and more than 41 publications in referred international and national journals. He has also supervised 5 students currently working under his guidance. He worked as an ICMR fellow and has received also worked for DBT, Ministry of Tribal Affairs. Many publications as in corresponding and first authors in high impact factor journals having impact of almost 6.1. And to name a few, to name his experience, he's worked as an currently working as a lecturer in JS College of Pharmacy. He has worked also with PhD College of Pharmacy, Coimbatore, as an assistant professor from 2018 and 19. He was a visiting faculty in September 2014 to 18 at Vasan Institute of Ophthalmology and Research, Coimbatore. So, with this brief introduction, I would like to invite Sir to start with the session.
Thank you, sir. And it's been a pleasure for me to share my experience with all the participants. I thank School of Pharmaceutical Sciences, Sandeep University, for giving me this opportunity. And today I will uh, give you all a brief introduction and idea about network pharmacology, which is one of the emerging field, very important in drug discovery process. So I will switch off my video for better clarity. So I hope my page is visible to everyone.
It's visible, sir. If it is possible, can you maximize it, sir? Can you full screen it?
Okay.
Yes, sir. Perfect.
Thank you. So this session is all about network pharmacology, its basics, methods, and applications in drug discovery. And network pharmacology has a variety of uses. Okay? But in this session, we will stick to how network pharmacology is used in especially drug discovery process, which is more useful as a pharmacologist and medicinal chemistry. So these are the topics we are going to cover. So first I will teach you basics about the network pharmacology. And the second one, what are all its applications, including drug discovery, and what are the other applications as well. Then I will give you a brief introduction on the how to do it, how to do the various applications, and I will give you a brief introduction on the methodology. And methodology, I can't explain it completely here, but I will give you some few papers of mine which you can go through it, where the methodology for each and every application is explained very nicely. You can use that. So first, starting with network pharmacology introduction. So it is a British pharmacologist Hopkins is the one who termed the word network pharmacology. Here, he used this word network pharmacology because he thought that once a single drug may affect multiple targets to bring out the therapeutic response. So this is the reason why he termed that as network pharmacology. Before that, this network pharmacology is called as network target, where uh, this word network target is used to um, mention the drugs which have multiple targets. And from that network target, network pharmacology have emerged. And it is a derivative of systems. Network pharmacology is a derivative of systems biology. System biology is an interdisciplinary field which helps us to model the complex biological systems. Uh, it uses mathematical and computational methods to build uh, a mathematical network for the complex biological system. And this system biology is the basics for network pharmacology. So this systems biology build the network of the complex biological system from the various omics database, omics data. Uh, we all know that omics or the omics data are derived from various high throughput technologies, where the high throughput technologies uses the ohm of our living system, like genome, various ohms of our living system, like genome, proteome, metabolome, interactome, lipidome. So from these data, this system biology helps to build a mathematical model for the complex biological system. So this model is used by the network pharmacology for identifying the various biological aspects of drug and diseases. We will come to know one by one in the coming slides. We will know what are the applications of the network pharmacology one by one. Okay, and this is a important word we can use for network pharmacology is it is an holistic approach. It doesn't believe in one target, uh, one drug, one target mechanism. So up to now, our most of the drugs have lacuna that it acts on only one target, or we use one single drug to target one particular pathway, which is not going to give a fruitful results in a complex diseases like cancer, diabetes. It won't give a fruitful disease because those diseases are outcome of a whole network, outcome of a various proteins. For example, cancer means it is just not mutation in one protein, it is mutation in many proteins combined together, okay, that relieves the break in the cell cycle. Hence, one target, one molecule approach for the drug discovery, for the curing of disease, it is not, uh, it has some lacuna, and that's why we don't have a very good drugs for complex diseases, which is actually opposite to the reductionist principle. So this is the network pharmacology. So network pharmacology is derived from systems biology. It uses the mathematical model created by the system biology for the application of drug-related process and disease progression. So here, this network pharmacology is an interdisciplinary field. It uses data from 4 different branches. One is pharmacology, one is bioinformatics, and another one is systems biology. And you need to create a model for it, including all these 3 systems. For that, we require network analysis. And generally, network pharmacology employs graph theory to calculate the data, to form a network, okay, for our interpretation using this network analysis, which we call it as graph theory. This network pharmacology has 2 important concepts. One is disease mechanisms are best targeted by multiple drugs that work together. This is one of the important concepts. Second one is effective drugs may work on multiple targets instead of one. So these 2 are very important concepts of pharmacology. Hence, since it doesn't see the biological system as one single, it sees the biological system as one single entity. That means if you take a cell, it 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 merges all the pathway in the cells to perform the basic biological activity. Hence, it doesn't consider, uh, it doesn't take apart. It actually it doesn't take apart the mechanisms which are happening in the cells. It helps to piece together, and it brings the whole mechanism as one system. So this is since it is whole lots of pathways are involved here in a cell, and it sees network pharmacology sees as that whole pathway as one entity. This is called as holistic approach, which considers all the the total living organisms, the interactions that happens in the living organisms as a single entity. Hence, it is called as holistic approach. And it became since it is considers the, how I can say, the functions as one whole. Okay, what happens here is it became an immediate hit with the researchers who are working on traditional medicine. Researchers who are working on traditional medicine. So here, what happens here is, we know in traditional medicine, it is not based on the principle. It is not one phytochemical that is present in an extract or a formulation, and all the phytochemicals work together to elicit its mechanism of action or the intended therapeutic effect. Since this traditional medicine, there are various phytochemicals are there, it is very difficult to uh, summarize the overall mechanism of action. And for that, this network pharmacology has become an important tool to propose a combined mechanism of action of the herbal formulations. It also helps to give an evidence to the effect, through miming the various databases which we use in network pharmacology. So these are the four pillars of the network pharmacology, because we need an output result in the form of graph. Okay, and that graph, from the graph we will come to a conclusion which are important, which are most important, which are least important. So here, if you see, this network pharmacology takes data from, as I said before, from systems biology, from the various omics databases, as well as pharmacology and bioinformatics. So the biology, bioinformatics, systems biology are combined together to form the network. To form a network. This is the network, okay? To form a network. So this is that simple diagram of a network. This network consists of two things. One is node and another one is edges. Just a moment. One is node and another one is the connection between the two nodes is called as edges. So these nodes would be the biological entities like our drug, our gene, or the protein. And this edges represents what type of interaction between the edges. It could be an inhibitory connection, or it could be a stimulatory connection, or it could be just a chaperone protein, or it could be a flow of information from one node to another. There are there could be a various association between a two protein, two gene, or between a drug and a protein. So this edge represents the association between the two nodes. Okay. So this node is a biological entity. So once again, I will repeat this because it is very important. The network pharmacology, it's a data. All are data driven science. Okay. It's a data. We get it from three important fields. One is system biology, second one is pharmacology and bioinformatics. So these three systems combine us to give a network. It could be a drug protein network, or a protein network, or a disease gene network, any one of it. Okay. Uh, with the help of network analysis, which in that network analysis we call this as graph theory. Okay. So final output will be like this. So you have a network with node and edges. So the nodes are the one which are the biological entities. It will be our drug, it will be our gene, or it could be our protein. And edges will be there. So edges represents the connection between the nodes. So these nodes have these edges with the connection between the nodes, and it indicates if experimental evidence are there, if a computational interpretation is done, then it can give us what type of connection between the nodes. So in a graph, okay, in a network, you will get an output like this one, This is called as network, and from this network, we can identify which are important nodes, which are prominent nodes. Then our research will be based on those prominent nodes. I'll explain it further now. And another one important thing is I have said before, the omics data, okay, from the various omes in our genome, proteome, metabolome, lipidome, or interactome. So we have several databases for these omes. The databases are created through some high throughput technology. For example, there will be a database for a biological pathway. where it says how the proteins, how the genes are expressed. And there will be another database where what are the genes were altered in the diseased environment. Okay, so with these, from these databases, okay, it will take the results and integrate it, and it will give us the interactome diagram, which we call it as network. So that's it. Next is also, these are the, as I told you, the omics approach. We have genomics, proteomics, and the metabolomics. The data will be taken from here for the construction of the network. Now, once a network is constructed, it is very important for us to identify which is important prominent node among the network. So for that, we will use centrality measure. We will measure centrality measures, okay? And we have three centrality measure. Important ones are, many are there. Generally, I will use three. One is degree centrality. Second one is betweenness centrality. Third one is closeness centrality. So what is meant by degree centrality is, in a, for example, this is the network we have got. This is the network we have got. In the network, we have, you can see here, we can, four clusters are, sub clusters are there. Here you can see 1, 2, we have a small cluster 3 and 4. Okay. So if you see here, in that cluster, which node is having the highest connection? That we call it as degree centrality. So you can see here, this one is having the highest connection with other nodes. Similarly, this here has the highest connection with the other nodes. So these are called as degree centrality. Just a minute, I'll charge my laptop. Thank you. So which node is having highest connections, number of connections with the other node, which we call it as degree centrality. And second one is betweenness centrality. This is my favorite one. And most of the findings also indicates that the nodes which has high betweenness centrality can function as a important target for drugs. So what is meant by betweenness centrality means they are they are placed in such a way that, okay, they are placed in such a way that, so this is the one example for betweenness centrality, a node which has high betweenness centrality. If you see here, this betweenness centrality, this node helps in the flow of information for three subclusters, 1, 2, and 3. You have four subclusters in the network. In that, you can see 1, 2, 3, these three subclusters, 1, 2, and 3. The flow of information is dependent on the betweenness centrality. So now, you think, for example, if in a cancer, okay, if this pathway is having making the cells to grow, it contributes to the cancer proliferation to a high degree, then if you inhibit this protein, so node means it will be a protein or gene, okay, then if you inhibit this node, then you can inhibit the, possibly you can inhibit the whole pathway. That's why this betweenness centrality becomes a very important in as a drug target. So this betweenness centrality means nodes bridging different network clusters. So this high degree of connections is called as hubs generally, and betweenness centrality, here the information flows through this node, hence we call it as bottlenecks. Then you have closeness centrality. So this is one example for closeness centrality, this node, because it has the shortest path with the other node. It has the shortest path with the other node. Now these are all mathematics. How we can relate this to biology? So in biology, for example, if this is a network constructed for a disease, just it's an just imagine that this is a network, okay, constructed for a disease. And if you want to target the gene or protein which has high degree hits, so if you target this protein using a drug, using a small molecule, what happens here is this small molecule, if you inhibit or stimulate this biological entity, what happens is imme- i- immediately the node, the information from this node, uh, which uh, the information from this node, okay, is transmitted to the nodes which are directly connected to it, okay. In between a centrality, this between a centrality functions as the flow of information. So if you inhibit this gene or protein, okay, or if you stimulate this gene or protein, the information is transported throughout the network. That is why it is very important as a target. So the third one is closeness centrality. So again, if you inhibit or increase the activity of this particular gene or protein, what happens here is this information is, since it has the shortest path with the other node, the information is transmitted very fastly throughout the network. So this is how these are the different centrality measures in the context of biology. Then finally, using this network, we can also identify the important pathways these proteins are involved in, which we call it as pathway enrichment analysis. So this is very important, okay, because this is how we will get a result. And using this network, we need to interpret our results. And from there, we want to move on to our drug discovery process. So, and coming to the applications of this network pharmacology, you can see uh, it's highly interrelated. It is highly interrelated. Okay. So the application is initially it was a very, as I told you before, it was a very hit among the traditional researchers from traditional medicine, because it helps to summarize the mechanism of the whole formulations, okay. But gradually it has evolved, and it has many applications now. See, one important thing is drug discovery and development, in that it helps to identify the target of the drug, okay, as well as the important target in a disease. So in target identification, it has two important use. One is it helps to identify the targets of a drug, not one single target, targets of a drug or the network of targets of a drug. Plus, it also helps to identify the important important gene or protein which are responsible for the disease progression. Then it will help us to identify the pathways which are altered in the disease mechanism. So which pathway is altered, and why, and how, what are all the proteins involved in that. So which is overexpressed, which is not overexpressed, which pathway is overrepresented in the disease. All those things we can identify using disease mechanism elucidation. Then, as usual, polypharmacology, that is the multi-target approach. Okay, a drug will act on multiple targets. Then you have drug repositioning. It's a very useful to network pharmacology. It's a very useful tool for drug repositioning. From our own laboratory, using network pharmacology, we have repositioned two drugs, and we have published those results also. In the coming slides, I will explain to you how we have done it. Then, in the traditional medicine, it helps to validate the biological effect of the herbal formulations. It helps to identify the therapeutic use as well as mechanism of action. So these are uh, primary, how we can say, uh, applications of network pharmacology. Apart from that, it can also helpful in pharmacogenomics. It helps to uh, personalize it helps to derive a personalized medicine. But this pharmacogenomics and the personalized medicine we are not going to cover in this session. Okay, you can look through journals. Okay, as well as you can, if you are interested in personalized medicine, you can also individually contact me. I can help you with that. For example, in personalized medicine, what happens here is, so if you can, if for example, again in a cancer or something, okay, if you have the patient's expression profile, gene expression profile, okay, you can use that expression profile, okay, to identify what are the important targets, what are all the important pathways, and based on the important targets in the disease, okay, you can change the combinations of the medicine, and then you can have a therapeutic benefit based on the individual genomic profile of the patient. So what are the applications today in this we are going to see is, we are going to see how the, what is, how to elucidate the mechanism of action, how to validate the therapeutic efficacy using in the traditional medicine, okay, using network pharmacology. Then how we can do a drug combination optimization, okay, identify a new drug combination for for a disease, and predicting side effects and toxicity, target identification for disease as well as the drug, how to do a drug repositioning, then how understand the disease mechanisms, understand the disease mechanism. So one by one we will see how to do it. So we move on to general methodology. So these are the 5 important steps in network pharmacology. The first and foremost thing is, you need to collect the genes. You need to collect the genes for the disease. For example, if you're working on a disease, you need to collect the disease related genes. For that, we have various databases are there. You have DisGenet, Malacats, okay. DisGenet is in subscription. You need to subscribe to it. Malacats is free to use, and there are several various databases also. OEM, IM is there. I'll show you the individual databases where you can get the disease-related genes, okay, at the end of this class. Then you need to identify the drug targets. Drug targets can be again identified from various databases. You have a similarity ensemble approach to one database is that. Then you can just take it from PubChem compound, Chembl, Binding DB. If you want to predict it also, you can use Swiss target prediction. So all those things. So first and foremost things is, you want to collect the genes or proteins from these databases for a drug, or it could be for a or it could be for a herbal formula. Herbal formulation means you will have various phytochemicals, and for each phytochemical you need to identify, you need to collect the targets. Okay. Then the second, network construction. So once you have collected the genes, okay, once you have collected the genes, proteins, okay, Using that, you need to construct a network, interactome we call it as. Generally, it will be a protein-protein interaction network. Generally, it will be a protein-protein interaction network. For this, you have again various databases. One important database is called a string database, S-T-R-I-N-G, string database. From the string database, in the string database, you can have this, you can generate the network, and this network can be uh, transported from the string into various analytical uh, softwares. So this is called network analysis. Network analysis. So for the third step is network analysis. The protein-protein interaction network, the interactome which you have created, okay, that could be fed into many softwares are available. Many tools are web tools are available. In that, one important one is Cytoscape, one software, okay. Using that Cytoscape, and another in the Cytoscape, you have a plugin called CytoNCA. Using the Cytoscape and CytoNCA, okay, you can do a network analysis. Here, what is mean by network analysis means you are going to find the prominent nodes of the network. So once you have done a protein-protein interaction, you will get a diagram like this. You can get a network like this. You can see here you have a node as well as edge. Node is the biological entity. It could be a gene, protein, drug, and edge is the connection between the gene, between the two nodes. So using this, okay, it's a using this network, okay, by feeding this network into an appropriate software, okay, you can identify the prominent nodes using the centrality measures. As I told you before, you have three centrality measures. One is um, closeness, betweenness, as well as degree. So once it is done, you can take this important nodes. Okay, you can take these important nodes to for an enrichment assay. Either you can go for a gene enrichment or pathway enrichment. In gene enrichment, you will identify the important genes where it acts. In the pathway enrichment, you will identify what are all the prominent pathways, what are all the pathways which are overrepresented, which can function as a key pathway for the drug action or for the disease. So here, if you see, for pathway enrichment and for gene enrichment, again, you have various databases to do it. So one of my favorite is Reactome databases for a pathway, and Go Ontology database for gene enrichment. So finally, once you have all done this, okay, okay, uh, you will be having couple based on this slide. I'm saying you will be having couple of results. One is an important target in the disease, or two or three targets in the disease, and an important targets of a drug. So the final thing is you need to validate your predicted findings. You need to validate your predicted findings using suitable in vitro, in vivo experiments. For example, okay, if you find that this protein is important in the development of this disease, then you can do a knockout study, or you can silence the protein using an siRNA, and then you need to measure the response, whether the disease is progressing or not. And if you find one compound which can act on multiple targets, so you need to do proper enzyme inhibition assay or proper in vitro assay, in situ, in vitro assay, and then you need to prove it whether it really acting or not. So the validation part is very, very important. I hope you can understand from the first set to fifth set. So first set is data collection. You are going to collect the genes that are responsible. Either you are going to collect the genes that are responsible for that disease. You are going to collect the genes, proteins that are going to act as a target for the drug. Okay. Second one is using that collected genes, you are going to build a network using a database called string. From the string, okay, you can export that data to various softwares and web tools are available. One of my favorite is Cytoscape. Using Cytoscape, you are going to identify the prominent nodes, prominent biological entities among the network. Because, for example, if you take a disease, if you take, for example, if you take a Parkinson disease, and there will be more than 1,500 genes will be there, which are responsible for Parkinson disease. Okay, so we need to identify the best one based on the network analysis. So how we will identify the best one? We will use the we will make a do a centrality measure, and during this using the centrality measures we will find out the prominent nodes. So the three centrality measures which we use is one is closeness, betweenness, and a degree. So once we found out the topmost uh, topmost prominent biological entity, using that we will do a pathway as well as gene enrichment, and finally we need to do a validation study to prove it. So without validation study, all these first four steps will become you can't prove it. The results actually may be there or may not be there. That can be only done using a proved using a validation study. So one by one, we will do how now. This is the general flow how to do a network pharmacology. Now we will do one by one. We will take. We have seen 6 applications in drug discovery process. For each applications, I will tell you how you can do it. So overview of task and networks in network traditional medicine validation. Here for identifying the mechanism of action of a traditional medicine, you can use this method. So what you can do here is first you can you will have an evidence already. You will have an evidence that this formulation or this plant is it is used for this disease in our traditional medicine. So here what you can do is, for example, if you have a disease X, okay, disease X and a plant Y or a formulation Y. So for this disease X, you can take the genes which are responsible for the progression of the disease, which are responsible for the progression of the disease. From various database, you can say, as I said, Disgenet, Malacats, OMMM you can use, okay? And you can, using various databases, you can various, how I can say, databases for plant phytochemicals. You can first you need to find out the phytochemicals that are present in the plant. For that, you can use the database called IMPAT, IMPPAT. Okay, generally I use it. And for the phytochemicals, you have variety of phytochemicals. For example, at least if you take a plant, the prominent at least 20 to 25 prominent phytochemicals will be there that you need to identify using a literature survey. So once you selected the phytochemicals, then for each phytochemical, you need to identify the target. You need to identify the biological target. For that, you can use many websites, many databases are there. My favorite is Similarity Ensemble Approach, okay, SEA. This is one of the website I will use. Apart from that, you have PubChem Compound. In that biological activity, you can see the targets. Then you have Binding DB, then Campbell. All these databases you can use to fetch the targets for the particular phytochemical. Now you have a list of phytochemical, as well as for each phytochemical you have different targets. Then you just pull the targets together, and then you have a genes, as well as here you will have the genes of the protein targets. Now you identify a common gene using Venn diagram. Okay, so once you have identified the common genes between the disease and your phytochemical targets, okay, common genes or proteins, okay, between the disease as well as your phytochemical target. You have a set of genes, okay. Using that set of genes, you can create a network using string, the same workflow. You can create a network using string, and that string, okay, that the network created using the protein-protein interaction which have created using the string, then you can feed that data into Cytoscape, and using CytoNCA plugin in the Cytoscape, okay, you can rank the prominent nodes using our centrality measures. Now you will have a idea about what are all the major targets these phytochemicals act on, okay. Second one is, with the same common genes, with the same, how we can say, common genes between the phytochemical, between the herbal formulation and the disease, you can identify which pathway it works on, and you can also do a gene enrichment study. Again, it helps to identify the uh, genes which are overrepresented. Okay. Pathway enrichment also same. It identifies the pathways which are overrepresented. Hence, uh, the biological activity of the herbal compounds might be mediated to the overrepresented gene as well as the overrepresented pathway. And finally, you need to prove these things using a validation technique, in vitro or in vivo validation technique. So this is how you will do a you will do a this is a basic workflow to identify, to validate the mechanism of action of a traditional medicine. Then optimization of drug combination. You can also identify a drug combination. If you see, this is the network which I have done it for essential hypertension. And what you need to do is, first you need to create a protein-protein interactome for whatever drugs you are going to test. It could be a full FDA approved drugs. Okay, so drug A, drug B, and the interactome, the genes responsible for the disease. So now you are created a, for here I am saying about 3 examples, but you can use how many drugs you want. Okay, so drug A, you are going to create a protein-protein interactome. Then for drug B, you can create a protein-protein interactome. For disease, again, you are going to create a disease gene interactome. So this is the interactome of the disease. Now you will have an individual interactome for drug A and drug B. So when you superimpose this the interactome of drug A and drug B, you can see here drug A, can see here it acts on this TLR4 pathway, interleukin inflammation pathway. And then you can see here, and for example, if another drug acts on this network, okay, this subcluster, okay, which is based on angiotensin, which is a very common thing which is a common target that the drugs will target in the the drugs which target this angiotensin in the for the treatment of hypertension. But here you can see, for hypertension, it is also mentioning this inflammatory pathway is also essential. So now you combine, you can test a drug combination, one from this angiotensin inhibitors like our losartan, enalapril, angiotensin converting enzyme inhibitors, which can inhibit this network, and another anti-inflammatory drugs that can inhibit this network. Then you can check this combination in vitro and in vivo, and then you need to prove it. Okay? So this is how you can find a drug combination where you will create a network for the disease first. Okay, then you will create a network for individual drugs, and you will try to superimpose the network of the individual drugs with the disease, and which is having the most matching one among the within that disease network, you can try these combinations. Okay. And the third one is prediction of adverse effects. So here adverse effects, again, if you can take a drug, okay, you need to create a protein-protein interactome, protein-protein interaction network for the drug. And you need to check, okay, you need to check whether here you are not going to use any diseases, okay, you are just going to take all the proteins which are found to interact with the drug. Then you are going to again you are going to build a network. Okay, from the network you need to do a target analysis. So the first step is you are going to choose the drug of interest which you are going to work. Okay, then you need to build a protein-protein interactions. Okay, this protein-protein interaction should be built for all the targets which have proven to interact with the drug. Okay, then you need to do the target analysis. In the target analysis, if you find proteins which are involved in multiple pathways, which we call it as hub protein, which has lots of connection with other nodes, then if you find a protein, okay, in the target analysis, if you find a protein that are bottlenecks, that is very, very important in the flow of information for various other biological pathways, then those drugs are bound to produce toxicity. Since you can avoid the drug at the initial stage itself, or you can predict the adverse effects of the drugs. Second one is again pathway enrichment analysis. So here again using this uh, genes which were collected for the targets which were collected for the drugs, okay, can be fed into a pathway, and then we can pathway enrichment analysis tool, and using that we can identify whether the drug is going to affect any important biological pathway. If it does, then again the drug may produce some serious adverse effects or toxicity. So these are the things how we can use prediction of adverse effects. But prediction of adverse effect with network pharmacology is still in very early stage. Okay, if you have a novel drug for the prediction of adverse effects, there are whole lots of online tools are available. Okay, ADME property prediction tools are available. You can use that. Then the fourth one is a target pathway identification. As I told you before, two targets you can identify. One essential for drug targets, and another one is for disease targets. So for a drug, how you can identify the drug target means, as we have, it is similar to what we have discussed in phytochemical, but in in herbal formulations, herbal formulations we have lots of phytochemical, and for each phytochemical we need to find out the uh, target. For each phytochemical we need to find out the target, but here you will have maybe one phytochemical or just one drug. Okay, so for that drug alone you need to comprehensively find the target. You need to find the target using several databases, as I said you before, PubChem compound. If you want to predict it, you can use Swiss ADME and then similarity ensemble, Swiss target prediction and similarity ensemble approach. And similarly, the disease interest, the disease genes should be collected from either DisGenet, Malacats. Then you need to find the common genes using a Venn diagram. So from that common gene, you can build a protein-protein interaction network. Again, this network can be fed into cytoscape to identify the prominent nodes. Those prominent nodes are your drug targets. Second one is uh, you can also identify the pathway using this. For that, this common genes, the list of common genes can be fed into a pathway enrichment analysis tool. The most famous is Reactome. I will use Reactome. KEGG also you can use. From that, it will give you the results for the results of the pathway in which the genes are involved. So by analyzing the results, you can also identify which pathway the drug acts on. And finally, again, you can do a gene enrichment to find out the important proteins which are responsible for the drug activity. So through these things, you can identify the targets of the drug, not one single target, targets of the drug. The second one is to identify the disease target. For example, if you have a disease and you need to find a new target to work on, simple. Again, you can collect the genes from the various databases, cystinoids and malacats. And instead of identifying the common gene, you can directly build the protein-protein interactome using string, and you can fed that. You can send the data to the cytoscape using cytoscape as a plugin cyto NCA. You can predict the prominent nodes. So those prominent nodes are your disease targets. Then further, you can validate the disease targets whether it is really important using some biological activity, and then you can prove it. Similarly, the disease genes, you can feed it into a pathway enrichment analysis tool, and then you can use this data to check whether which pathway is uh, overexpressed, which pathway is overexpressed, and you can, so that you can use take that results and you can target that pathway to overcome the disease burden. So drug repositioning, this is one of the highly used method for repositioning the drugs. So what you are going to do here is you are going to build a protein-protein network using the drug and all the same procedures you do, and then you find the prominent nodes. That means the prominent targets. Okay, using the prominent targets, you have multiple targets, right? You will have multiple targets. So you can manually check whether that target is also represented in other diseases. For example, if you have a target X, and if the target X is also involved in other diseases also, then you can test this drug to the in that another disease where this X, the target X is present. This is a very simple technique. I'll give you an example at the end of the slide. Okay. Then I'm understanding complex disease mechanism. Here, there is a one technology called array technology where you can study the various expression of the proteins and genes in a tissue. For example, is very useful in cancer. Okay, what happens here is if you can take a cancer tissue or cancer cell, and then you can do a expression profiling of the uh, gene or the protein expression profiling in the cancer cell. Now you will have a set of genes which are expressed. You can compare with those genes in the normal tissue. Now you have a result of which is overexpressed and which is underexpressed. Okay. By building a network using this normal expressed proteins as well as the in the normal tissue as well as the expression involved, the expression of the genes in the cancer tissue, you will have two contrasting networks. You will have a two contrasting networks. Using the contrasting networks, you can find out which pathway, which genes are disrupted in the disease, and using that you can build a model so that you can particularly target the disrupted pathways or the disrupted genes. So finally, experimental validation is very, very important. Okay. Because even though the data for the network pharmacology is taken from there, how we can say most of the data are from experimentally validated procedures, especially high throughput technologies. But few of the data are also computational and extrapolated. So validation becomes utmost important. So whenever you do, you need to have at least a basic validation study, like a small efficacy study. For example, if you think that if you find out this one enzyme is very important, then you need to do an enzyme inhibition assay to prove that mechanism of action of your compound. And if you have a receptor, then you try to do a radio labeled binding assay or a transfection assay to prove the your drug acts through the receptor, at least an in vitro and in vivo mechanism. Okay, and measuring the expression of the genes which you had predicted. All those things are very important. Okay, so validation techniques becomes very important in network pharmacology. So as I told you before, these are the web tools and databases used in. So I'll go one by one. So for target identification, that means if you have phytochemical and drug, and you need to identify the target for this, you have Swiss database, Swiss target prediction. You have binding DB, you have Stitch database, and you have a similarity enzyme, lots of PubChem compounds. So all these databases you can use to identify the fetch the target. Here, the Swiss target prediction is the predicted one. Okay, it has both predicted as well as proved one. So whenever I use, I will avoid Swiss target prediction, and rest all have a quantitative value for the interactions, either for inhibition or activity. So I will use the other databases like binding DB, Stitch, C, and PubChem compound. C and PubChem compound is my favorite one based on my experience. And then second one, disease genes. So if you want to take the genes which are associated with the disease, dysgenet and therapeutic drug target database, Malacats, OMIM gene codes. Here, recently, dysgenet has become now it is only available as a subscription mode. Okay, so dysgenet is the best one, but since it is available in the subscription mode, you can opt for Malacats, which is also it's a good one. Then for selection of common genes, you have a website. Okay, either you can use the Excel or you can just use this website, Veni 2.1.0. So this is the link for that. I will give the slides to the organizers so that participants can get these slides and then you can work it on. And for protein-protein interactions, generation of protein-protein interactions, I will use the database called string database. Okay, and for the identification of the prominent nodes using centrality measures, I will use the Cytoscape software. Okay, it's free of cost. You can download and then you can use it. String, it is also free, but it is a web server. So then pathway and enrichment analysis. For pathway enrichment analysis, my favorite thing is reactome. So this reactome gives the biological pathway, as well as there are other pathways are also there. One is KEG, but that will give you more of disease pathway and not of a biological pathway. Then you have for gene enrichment, you can use Clugo, ShinyGo. My advice is use Go ontology. Okay, my advice is to use Go ontology. Then at finally, so now all those network pharmacology are there. Now, how it helped in the real world. So network pharmacology helped the scientists to identify these two proteins, these two genes or proteins. Okay, as a critical hubs. It's a critical hub for the treatment of cancer. For the treatment of cancer, again in Alzheimer disease, it help to identify the critical proteins that are involved in the clearance pathway. the amyloid beta clearance pathway. And in COVID, it helps to identify the two targets, okay, which are vital. We know this two angiotensin 2 and TMPRSS2 are vital targets for interactions with the virus, okay. So in real world, uh, these are the in their world, it helped to identify key targets like EGFR2, HER2 receptors for the treatment of cancer. In Alzheimer's disease, has helped to identify the important proteins in the clearance pathway of the amyloid beta. In COVID-19, helped to narrow down these two enzymes which were responsible for interacting with the virus. And in the identification of the mechanism of action, okay, this network pharmacology helped us to identify the multi-targeting ability of sorafenib, okay, which can inhibit both angiogenesis as well as tumor proliferation by targeting various kinase receptors. So this is our real world examples, okay, which are in the market, which we have employed in the humans, okay. Now from our laboratory, what are all the work we have done using network pharmacology? So this helped us to identify, one is repositioning, okay. This network pharmacology helps us to identify ropinirole for the treatment of Alzheimer disease, okay. So in this study, what we have done is we have used a hypothesis that the drugs which have similar side effects, okay, may also have similar structure, and hence it act on the same biological pathway. This is the hypothesis we have kept. And based on this hypothesis, what we have done is we have took all the drugs from which are all the FDA approved drugs for the Alzheimer disease, and then we took all the we aggregated all the side effects of the drugs, okay. And we have a software web tool called Cider where you can do it. And another one website is there that is called the Promiscuous, okay, where you can get the side effects of the drugs. Then we matched that side effects with all the FDA approved drugs. So whichever is having highest matches, okay, we initially done an in silico screening and in vitro screening, and based on that we found that this ropinirole had side effects that matches with the already existing FDA approved drugs. So we thought this ropinirole could act on the same biological pathway like the drugs which have been approved for Alzheimer's disease. And we have done a study with that, and we got a positive results with ropinirole and some of the targets. And using that, we have published a paper, okay, as well as we thought that some of the targets where ropinirole binds has a potential to increase its affinity. Hence, like similar with the same ropinirole, we have made some derivatives, and then again we checked their activity in silico, and we got good results. With that, we applied for an ICMR project, and we have received the grant for the same using this network pharmacology method, which started this work. Then the second study is here we found that the plumbagine could be used for using literature survey. We have found that plumbagine, a compound, okay, actually it's a toxic compound, could be used in treatment of Parkinson disease. But when we typed plumbagine in the literature, we retrieved around 900 hits. So 900 hits means plumbagine is very highly explored. Now we want to understand what is the mechanism of action in the plumbagine in Parkinson disease. For that, we have used applied network pharmacology, and we found out that these the plumbagine may work on these mechanisms in Parkinson disease. and we tested that tested using some efficacy studies, and we have published that paper in a molecular diversity, which is again a very good journal. Okay, and then another one here. Another one of my students was very interested in working on uterine fibroids, and what we have done is we thought of working we thought of trying a repositioning work. So here what we have done is, first we have studied about uterine fibroids, and then we found that one of the protein called a fibroblast activation protein, which is responsible for the creation of the fibroids, the excess amount of extracellular matrix materials in the uterine fibroid, and we thought to inhibit it. So instead of going for a screening, a molecular docking work, what we have done is we used a database called Stitch database, STITCH, Stitch database, and then we fed the information regarding the fibroblast activation protein, and it has given us several compounds which has possibility of interacting with that. In that, we found this our DPP4 inhibitors, linagliptin, vidagliptin, penagliptin, all are having inhibitory interactions with the FAP. So what we done is we thought to evaluate its mechanism, evaluate it in vitro and in vivo to prove it. Then we have used all telegliptin, vidagliptin, langtin. In that, we found linagliptin has the ability to decrease the uterine fibroid formation. And with that results, we have published one paper in biochemical and biochemical and biophysical research communication. Now, another one example is this one. Here, one of my students was interested in working on metabolic disorders. So what we thought is we can work on type 2 diabetes mellitus. But type 2 diabetes mellitus, if you see again, there are only the targets, the molecular targets for type 2 diabetes are very small. And again, it is PPR, gamma, this sulfonylurea receptors, only few are there. So what we thought is we need to come up with a new target like that we have got. For that, we have identified the target. What are the targets, prominent targets in the type 2 diabetes mellitus using some same network pharmacology. In that, we got one peculiar result, that is androgen receptor. Okay, and we thought of pursuing with androgen receptor, and we found some evidence that testosterone might be very useful in treating type 2 diabetes mellitus among males, and we got the androgen receptor results as one of the important targets. So with this basics, what we have done, we try to identify novel androgen receptor modulators for treating type 2 diabetes. With that, now we have published a couple of papers on hypothesis, and another one research paper are published. And ICMR also granted a fellowship for performing the study. And in conclusion, uh, it is a you need to the dimensions should be changed. Okay, the drug discovery process, the dimension of the drug discovery process should be changed to treat complex diseases. Okay, for example, if it is an anti-inflammatory drug, COX inhibition is sufficient to reduce the pain, but in case of cancers, in case of complex diseases, in case of Alzheimer, in case of many neurodegenerative diseases, where complex mechanisms are involved, you need to change from single to multi-target approach. That is very, very important. So this is the concluding remarks of my presentation. And challenges, these are the challenges. First one is still we have a limited data, and the data integration and quality, there are not lot of methods are available to validate the results which we get. Okay, so data integrity is very, very important. Data integration as well as the quality integrity of the data is very, very important. and we need to use various methods to predict the accuracy of the data which we will get. Okay. As usual, one of the important problems to address is the complexity of our biological environment. It's very difficult to, how we can say, uh, it's very difficult to piece together the complex nature of the biological activity. And finally, confirmation of the predicted results is ultimately important. Thank you. Thank you, Jackson.
Yes, sir. Thank you, sir. Hello?
Yes, sir.
Yes, sir. Thank you. Thank you so much for your fruitful session. Dear participants, now the session is open for questionnaires. If you have any doubts, you can ask.
Hello, sir.
Yeah.
Yes,
Am I
sir.
audible? Yeah. For this wonderful session, sir. I have one simple. Hello.
Hello?
Yes.
Yeah, you continue. Continue.
Hello.
Yeah. I can.
Yeah,
It's audible. You can ask.
Hello?
hello.
Yes, Vijay, sir. You can continue. You can ask the questions.
Okay. Thank you. Thank you, sir. Sir, I have a question. You said reactome pathway and cake pathway both are very, and when we prepare a network, right? So in that
Yeah. Vijay,
any of
I
them,
couldn't.
either.
No, I couldn't hear you properly.
Okay. Now is it?
You can
Now am I
type.
audible?
Yes, sir. Yes, sir.
Actually, you are.
Now you're audible. You can speak now.
Yeah.
Okay. So what I was saying, there are reactome pathways and there are cake pathways, right? For network preparation, we can use any of them. Okay.
Please,
But the question,
okay.
but the question arises that when we are using cake pathways, right? So it would show more than 20, 20, 50. It depends on the number of this proteins we are having in our network. But once again, it will show too many pathways. Now, which pathway we should select and which pathway we should avoid? That would be very important to query sometimes. If you can explain on then at that point something.
Actually, each pathway it will be ranked on various criteria. One is the p-value of the pathway. Okay. So if we all know that a p-value which is less than 0.05 will be the validated one. So you should select the pathway based on the statistical parameter. For example, a reactome and as well as KEGG have a different statistical method to predict the confirmation. So if you go through those papers, you will find that what are the statistical method they have used. Generally, there will be a p-value for that, and that p-value which are less than 0.05, you can select.
Okay, okay, sir. Got it, sir. Thank you so much, sir.
Okay.
Hello, sir. I have one doubt. This is Ronak here.
Yeah.
So, sir, currently I'm focusing on a one metabolic disorder that is non-alcoholic steatohepatitis relating to the metabolic. It is a metabolic disorder, type 2 diabetes primary reason. So,
Okay.
how can network pharmacology help me in selecting the targets and validating the targets?
First, you need to collect the genes which are responsible for your particular metabolic disorder. So once
Okay,
you
sir.
have collected the gene, then you can build a protein-protein interactome for that gene using string database.
Yeah, sir. That gene collection I have done from the database is different. So for
Okay.
DSG Net, I have used Metascape platform. Is it
Metascape.
correct?
Okay. Metascape is
Whether
also
it is a
fine.
correct? Okay.
No, Metascape is also fine, but the problem here is, uh, if you want to identify a target to work on, if you want to identify a prominent node to work on, it is better you create a network using string and then use Cytoscape or Network X. Another one app is there to apply the graph theory to for the selection of the prominent node. That will be better.
Okay.
Yeah.
So no,
Metascape also we
I
can
have
use.
used
Yeah.
three databases, that is GeneCards, CTD, and this one, string database. So from that, I
Okay.
have chosen mapped targets,
But here,
mapped also.
but okay, but here what you have not used is that you didn't use the graph theory up to now. That is very important actually.
Graph theory, sir.
Yeah, graph theory that can that that can be the graph theory will give you the prominent nodes. To find the prominent nodes, you can use that software Cytoscape. That is very important.
Okay, graph theory, sir.
Yeah, graph theory. Yeah.
Okay, thank you, sir.
Mm.
Yes, any more questions from the participants? Yes. Thank you, sir. We are concluding the session by the few remarks. Thank you, sir, for your fruitful and wonderful session. You gave the step by step how we will do the network pharmacology and what is the applications of network pharmacology and what you done the research in your lab, like for Parkinson's and uterine fibrinoids. It's a wonderful research work. So, dear
Okay, thank you.
participants, if you want any collaborations or any suggestions from this sir, you can contact with sir. So, sir will always help you without any hesitation. So, thank you, sir, for your acceptance as a resource person for this online training program. So, we hope we will collaborate in future also for the coming events also, sir. Thank you, sir.
Thank
And
you.
this is the token
Thank you.
of appreciation for the certificate. Yes, sir. Thank you, sir. Thank you for your wonderful session.
Thank you.
Thank you, my dear participants. We will meet you tomorrow at 10:30 with a new session. Thank you.